So, thank you. Yeah, so my title is The Interdependence of FMDV Pathogenesis, Challenge System and Outcome of Vaccine Studies. And in a more simple translation, that's just something about how the study design and specifically the challenge system matter for what we could and should conclude from our vaccine studies. And this presentation might be a little bit different from most of what we've heard this week, because I'm actually not presenting any experimental data or any new techniques. This is more of a summary of observations that we've done through a large series of animal experiments, with a little bit of addition from some other published examples. Also, since we're at the last day, I suspect others like me are a little bit exhausted by information overload, so we're going to start really simple with some basic bovine anatomy, because there are some aspects here that I believe directly affect FMDV pathogenesis and that often get either forgotten or just neglected when we're doing experimental studies. So this is kind of back to first year of vet school for those who've been there, and for those who haven't, this might just be something completely new and novel. But this is a cow's head, at least that's what it's supposed to be. And within the cow's head, you have here in blue and red the nasal cavity and the oral cavity that are divided by the hard palate, which in this cow is in purple. Within the oral cavity, you also have the tongue, which gets to be quite relevant when we start talking about FMD. The nasal cavity continues into the nasopharynx, and the oral cavity continues into the oropharynx, and those two are separated by the soft palate. If we go further back still, we have the trachea and the esophagus. Now there's a color coding pattern here, so if you follow the path of the blue shapes, that corresponds to the path of air that flows through the nasal cavity and the nasopharynx through the trachea down to the lungs. And in this relaxed breathing state, this soft palate is tucked in underneath the tip of the epiglottis at the opening of the trachea, so that you have this open path for air passage. However, when the animal swallows, things change quite dramatically. Well, they change. The tongue gets pulled backward, the soft palate is lifted, the epiglottis flaps up and closes off the trachea, so that you instead have this passage where feed can pass from the oral cavity down into the esophagus. Of course, these are three-dimensional structures. It doesn't actually look like this bridge that opens, but the main message is that there's this massive shift in the alignment of these structures. And why do we need to know this? Well, this thing of probank sampling is quite a big thing when we talk about FMD research, and for anyone who's ever collected a probank sample would know that once you insert this probank cup into the mouth or oropharynx of the animal, you induce a swallowing reflex. And by that, you're actually accessing the nasopharynx. And furthermore, if you've been a little bit unlucky with your probank sampling, you would know that you quite often get kind of stuck in there, and you have to be a little bit patient to actually get your probank cup back again. And that is what happens when this soft palate closes back to its neutral position when you have your probank cup in there. And a little bit of bonus information there is that we often do necropsies and tissue collection of animals that have recently been probank sampled. And when we do that, we often see this bruising or scarring actually on the dorsal surface of the soft palate. So you definitely do get there with your probank. Anyways, we're supposed to be talking about FMDV pathogenesis, and this slide is supposed to illustrate what happens when a cow gets exposed to FMDV via a natural route of exposure. And by natural here, I'm referring to anything that would lead to exposure of the upper respiratory tract, such as direct contact challenge. When we do that, we see this initial or primary virus replication that sets up within the nasopharyngeal mucosa. And if you're lucky to capture that with your microscope, it looks just about like this. I'm sorry for the people in the back who might not see, but the green color here is the epithelium of the mucosal surface, and within that you have these few little red cells that are virus-infected cells within the epithelium. Now, assuming that this is a naive and susceptible animal, this primary infection leads to a progression of events that we're quite familiar with. We get a local and systemic generalization of virus, viremia, the characteristic lesions. That's followed by an antibody response and clearance of virus. But in cattle, in a large proportion of animals, we see this persistent infection where the virus can still be found within the nasopharynx. And again, if you're very lucky and happen to collect the right piece of tissue, that looks just about like this. And again, the green is your epithelial surface, and the little tiny red cells that you almost can't see, but they're there, they are the virus-infected cells in the surface of the epithelium, and those will also be in your probank sample, so connecting the dots. So that was the all-natural basic situation. However, when we do experimental studies, we're not very natural about it. Specifically with vaccine challenge, we need everything to be standardized and stringent. So rather than using a natural route of virus exposure, we inject a rather large volume of virus directly to the tongue of the animal and set up this primary infection in the tongue. So by doing that, we basically bypass everything that is related to mucosal immunity and the nasopharynx. However, again, assuming that this is a naive animal, the only thing that really changes in the progression of pathogenesis is that we now have this primary infection in the tongue epithelium instead of in the nasopharynx, but we still have this otherwise similar progression of generalization and clearance and also persistence within the nasopharynx. But because of this generalization of virus, the virus will find its way into basically any tissue, including the nasopharynx, where it's capable of persisting. However, this changes if we use this tongue inoculation system to infect vaccinated animals, because then we have this strong primed antibody response that will prevent the virus from actually disseminating. And assuming that this is a very strong, presumably homologous response, this vaccine-induced immunity can actually effectively prevent the virus from reaching the nasopharynx or other tissues in the local area. So, and of course, this is kind of the extreme end of the scale where we have a perfectly homologous and strong response. There's a lot of gray areas where you may still have clinical protection, but you will have some local spread of virus. But we're sticking to the simple case here where you have a perfect protection. And what happens then is that you may or may not have some virus replication happening in the tongue, but you're basically canceling all other aspects of FMDV pathogenesis. There's no dissemination of virus. The virus isn't going anywhere. Of course, this is what you want your vaccine to be doing, but it might still not be a perfect replication of what would happen in a natural scenario because, that's my persistence, because if you were to repeat this, but with a natural route of virus challenge, you would instead still have your primary infection set up within the nasopharynx. You'd have the same primed vaccine-induced immunity that would now restrict the virus from going anywhere else, but since the virus is already within the nasopharynx, you will still have a proportion of animals that become subclinically infected and persistently infected. So in this scenario, you have the subclinical primary infection, and the virus may persist and cause subclinically infected carriers without any evidence of dissemination or clinical disease. That's the point. This is just an example of a real-life scenario where we have I'm not showing you any actual data, but this was actually two experiments that had a very similar design. Animals were vaccinated and challenged with the same combination of vaccine and virus. The only two differences were the challenge system. So in the first scenario, we had a tongue inoculation, whereas the second experiment, we used a simulated natural route of virus exposure, which in this case was intranasopharyngeal instillation of virus. The clinical outcome was completely the same. There was no clinical disease, no viremia. But in the first experiment, we didn't have any carriers at all, but when that was repeated, we had a prevalence of persistent infection of 90%, which was a bit different. And of course, there are multiple other sources of variation in vaccine studies. I'm just going to pull up a few examples from published literature. This first one by Fedra Eble and Aldo Decker's group showed a significant difference in protection depending on the timing between vaccination and challenge, and that's not that surprising. That's quite commonly reported by other studies too. But what I found was more interesting with this was that when the same group repeated a similar study, but they changed the challenge system to a direct contact exposure instead of needle inoculation, then the pigs that had been vaccinated 14 days prior to challenge were no longer protected. Because you could argue this thing that has been discussed here too, that this contact challenge of pigs might just represent a a much greater, larger, effective challenge dose of those animals, but it's a little bit harder to extrapolate from that which of those scenarios would be more a better representation of a field scenario. Similar but different, this is a paper by Sarah Cox and Paul Barnett's group from 2006, which very neatly showed that a tenfold increase in antigen payload could prevent persistent infection. This study was based on a contact challenge system, so nothing really to complain about there. In fact, we liked it so much that we tried to repeat it, but in our setup with a slightly different design, different virus and different vaccine, we saw absolutely no effect of increasing the antigen dose by tenfold. So my main message for this is that the way you design your experiment directly dictates what conclusions can and should be made. And specifically be careful if extrapolating information from a study that wasn't actually designed to answer that specific question. And as we know, that vaccine performance will depend on interplay of multiple factors, and the standardized models are a way of actually dealing with that. But I think we should be careful to not over-standardize our models. We heard about overfitting of data yesterday, and I think there's sort of a similar case that when we design our models, we should remember what the biology of this disease is actually like in the field and base our conclusions from how our model relates to that. With that, I'd like to acknowledge my collaborators and funders from the US government and University of Minnesota. And thanks to you for listening.
Okay, thank you. We have time.